Alrighty, guys. Hello, everyone. I hope you're having a good, relaxed summer break. Not too relaxed, I hope. Still trying to take advantage of prepping, for our last unit. And for these videos, or this video that I'm gonna make for you guys, it's essentially covering chapter 3, right? Sorry, chapter 13. The Unit 4 Biology Waste course, and we barely started getting into chapter 13, right, which is spread of pathogens, and this is probably the longest chapter in the waste program, right, or from the textbooks that we're using, which is spread of pathogens, right? We went over the life cycle of a pathogen, if you guys remember, the modes of transmission. And now we kind of left off the term, right, with the specific life cycles of pathogens, of some particular ones that you need to know. Remember, you need to know 10 diseases for for the waste program, right? That we're going into much more detailed in this section, right? So it's just something you need to learn, one way or another, right? You must memorize this. You need to learn it for the ATAR test, right? So I know it's quite extensive, but I think it's important that I'm actually recording this for you guys to have in detail rather than going over it in class and in a rush when we do have a limited number of classes, right? So the purpose of these videos is for you to get ahead, know the content well in chapter 13. When we come back, you guys can ask me particular things that you're unclear of, and then we can just get into chapter 14 and try to finish that chapter, and then the remaining of the classes you just review different question forms, practice some multiple choice for the ATAR test. Right, guys? So I don't want to spend too much time explaining here. And I do want to mention, you don't need to you don't need to see the whole video in its entirety all at once, right? As I talk about different pathogens, those are good places to stop, right? Or put a little pause. You can watch it another day, and so forth and so forth. So you won't get overburdened, right? But this will be quite an extensive video. If you notice, chapter 13.3, section 3 is this amount of panels or pages here, right? 10 pages. And then that's just the life cycles of the pathogens. And then the book gets into factors that affect the life cycles, right? Or the mode of transmission or the spread, right? Here you have 8 in general. So this chapter is quite extensive, at your own pacing. You can see every little section here, right? I do recommend, to go over each one. And then if you need a break, like for example, this one that's influenza, we talked about influenza already. That was the last thing we talked about. And then let's say I finish that, get into the Ross River virus disease, right? And then let's say you want to take a break, you can stop, for example, on sac brood disease, right? Or deformed wing virus. Every particular topic is a good place to stop, right? And so forth. So getting into the actual class content, right? In our last class, we talked about the Ross River virus disease, right? Or in other words, epidemic polyarthritis, right? Which was basically a viral disease transmitted by the mosquito vector, right? You get these particular symptoms. They're similar to arthritis, right? And there's particular species that are compatible with the pathogen to transmit it to susceptible hosts, to humans, right? which is the Aedes vigilax or Camptorhynchus, which is more particular to Australia in salt marsh and coastal areas. And then you have a more common widespread species, which is the Culex genus, right? I talked about a little bit of the anatomy. Let me kind of spread this out so you can see it better. A little bit about the anatomy of a mosquito proboscis, right? That there's 2 points of entries, right? Or there's 2 tubes within this whole structure, right? Which is called the proboscis. And one tube basically sucks in blood, and the other liberates saliva onto the skin of the host. Let's say like human skin, right? So ultimately, you know, the reason why we get mosquito bites is because, and they blood feed, is because, you know, they're in their reproductory stage that they need to feed to provide nutrients. They're producing eggs, and they need to supply these nutrients for the young, for the eggs to basically reproduce, multiply. Okay. Getting into the actual cycle of the mosquito and the disease, right? I want to come in here. I'll put this here. Okay, so one thing to note, right? This disease is infectious but not contagious, right? So it doesn't transmit directly from human to human. You basically go from from that mosquito vector to an intermediate host or a reservoir, right, onto a that susceptible host of a human, right? So the book kind of discusses it in a lot of detail in terms of paragraphs, but using this panel, this little diagram here, you could essentially get a sense of what's going on here. And I added some additional notes at each stage here, where we could essentially get some comprehensiveness, right, on how this disease gets transmitted. All right. So let's say you have a marsupial reservoir, in this case, like kangaroos, right? And they, let me see here. All right, let's start off at this point. And they're infected with the pathogen. They're infected with a virus. All right. So mosquitoes come in and they suck in, they blood feed, right, the kangaroos, right, and blood is coming in one direction from the marsupial, from the kangaroo into the mosquito. Okay, so there's a piercing of blood vessel intake of virus. Okay, and then now the mosquito has acquired the, if I can say acquired, has acquired the virus, right? The Ross River virus. And if the mosquito is the right species, there's a compatibility, right? Not necessarily only because you got the virus is going to immediately reproduce in you. There needs to be some form of specific protein level specificity where the virus can adhere to certain cell types, to certain tissues, right, where it can reproduce. So if the mosquito is the right species, the virus will tend to attach to epithelial cells, more specifically, you know, very specific receptors, ligand and receptor interaction, and that will trigger a replication process. Okay, so let's say you do have that that adequate mosquito and that adequate virus, right, and everything's continues on. Okay, so the virus is replicating inside the mosquito, and then the virus, of course, is spreading out, and it gets into the salival glands, and therefore the saliva that the mosquito is producing. Okay. Then what happens? The mosquito comes in and flies and does another blood feed on another species, another organism. In this case, you have humans, right? So, and again, remember, these are female mosquitoes. So you're going around to get another blood feed. As a mosquito, essentially you come into a human, you pierce with your proboscis on the human skin, right? But now instead of sucking in blood, whatever you get, you're liberating saliva. Remember that the mosquitoes have two tubes. You're providing saliva to what? If you remember, essentially to kind of serve as an analgesic, basically to numb the skin, so we don't even know that the mosquito is biting us. The saliva is being injected into humans, and that's where the virus is coming into humans, right? Not via the blood, but it's via the saliva. Okay. And as this is happening, again, similar situations, the virus is coming in, they can have multiple receptors for different species, as is in the case for the mosquito and epithelial cells. But in this case, right, the virus comes in and basically binds to receptors in muscle tissue, okay, in muscle tissue, and it starts replicating there, basically getting that period of incubation that we talked about, right? In this case, for this particular virus, you have 3 to 9 days incubation before you start feeling some form of symptoms, right? From this virus, right? So in the muscle cells, that's the primary replication area, okay? In the skeletal muscle cells of humans, okay? And then you have that susceptible host, which is us. It is possible, you have that mosquito vector, and you pretty much get infected, right? And that's how a human gets that disease, right? If a mosquito comes in and then bites or blood feeds on a human, right, it's not as likely to pass on to a marsupial vector or marsupial reservoir. Okay, it's always this kind of trend where you're getting these female mosquitoes, and then the new mosquitoes will likely get the virus from a kangaroo, right, rather than sucking in from blood from humans. Of course, they're not circulating the virus from the bloodstream because they lodge primarily in muscle tissue anyways, right? So this kind of circle that you see here, not likely, okay? Not likely. It may happen, but it's not the common form or the quickest, easiest form, all right? So it always starts essentially from the kangaroo up to us, okay? So this is essentially what you need to know in terms of the Ross River virus disease and its mode of transmission, okay? Where you have your reservoir, your intermediate host, and you have your actual susceptible host. All right, and so still talking about viruses, we talked about influenza, how it affects humans, Ross River virus disease also affecting humans, but not. Again, these are common ones that can occur in Australia. You do have some common ones with other organisms, in this case, honeybees, right? So many viruses can affect other organisms, and in this case, you have bees, and bees, of course, incredibly important because they serves as a pollinator, right? For flowers. Okay, and bees can get different types of diseases. We won't go through all of these, but I just want to name them. What are different types of diseases, right? So you have the sac brood disease, you have the black queen bee virus disease, chronic bee paralysis disease, deformed wing virus disease, right? Each of them have their own symptoms, characteristics, right? And in this case, we're only going to talk about the sac brood disease and the deformed wing virus disease. And again, as we're going along, I kind of want to mention, if you know the virus, basically and the effects that it does, then that's the likelihood of what the disease is called, right? Sac brood disease, deformed wing virus disease, right? Pretty straightforward there. So you have in terms of these viruses, the symptoms that bees can show could vary, right? Some can have very mild symptoms. Some can have very potent and very virulent disease symptoms, right? Some have no symptoms at all. So, but what's important is that those to know and understand, especially in research and science, right, is that those that are very virulent, their effectiveness of killing the hive is important to understand, because of course, if you kill a whole hive of bees, you're decreasing the population. The population gets decreased massively, right? And of course, there's a decrease in pollination. You don't get flowers reproducing. If you don't get flowers reproducing, that's a whole other trend in itself, right? That's a big problem in itself, right? So this is why it's important to understand how disease can affect bees. Okay, let me just move this here. Let's go to the continuing panel. So going into the specifics, not too detailed, right? In terms of bees here. So the first one is called sac brood disease, and it comes from another virus that's called the sac brood virus, right? And this virus is very broad spectrum, if I can say, in terms of the specific species, right? There's it can affect various subspecies of bees essentially all over the world, right? And more specifically, it affects worker larvae at the larval stage, so baby worker bees, right? But also adult bees can be affected, but moreover, it's the larvae that get affected, okay? And some symptoms with these bees get infected, right, is that they essentially turn upside down and they tend to extend their whole body, right? Moreover, the head portion. And what happens as a symptom, right, and as a kind of observable sign is that dead larvae, they tend to suddenly change coloration, right? From light to dark brown, ultimately turning into a very black kind of, in an extreme way, like a carbonous carbon shade, right? And they become very brittle. They break easily, right? And this would be an example of a beehive that has been affected by sac brood disease. And those little sites where you have, or each little individual honeycomb that contains these larvae, right, tend to get this very dark coloration, right, from this type of virus, ultimately killing a wide portion of worker bees. Okay. The mode of transmission, I didn't really bring put a panel here, but how the virus gets transmitted from one to another is essentially you have another group of bees, the nursing bees, basically, that they come in and they provide food and nutrients for the larvae. They're essentially contaminated. They're essentially infected, and as they're providing the nutrients to these larvae, they are passing down the virus to the larvae. So as they're kind of hopping down from one to another, they're infecting the whole situation, the whole group of bees, right? So the mode of transmission is basically coming from nurser bees that is spreading the virus through all the little larvae. Okay, so this is sac brood disease. The next one is called deformed wing virus. Okay, virus disease, of course, is called by deformed wing virus disease, and it's caused by the deformed wing virus. And symptoms could vary from being asymptomatic to severe. In that case of severeness, is essentially you're having a defected wing, right? You basically have no wing. You just have a certain kind of fibrous tissue there, or some of the main body location there. But but you're unable to fly, right? So that's why it's called deformed winged virus. And this has a vector, which is the mite, okay, the varroa mite, usually transmitted by the varroa mite, and it is a pest. The varroa mite is a pest worldwide, which can contain this virus, right? Interestingly, this mite itself has been very well controlled in Australia, right? So you don't see this that often, but you have seen there's some cases there. All right. So the actual scientific name of varroa mites is Varroa destructor mite species. You know, they could transmit this virus, having some similar form of transmission like mosquitoes, which is blood feeding. All right. This is a picture of the mite. Okay, and they're very microscopic, and they can lodge into the crevices, if I can say, or the folds of the body of the insect, right? They kind of lodge themselves in there, right? And if you have the virus, you know, and you're pretty much producing eggs and offspring, you can pass it down, as the case of a queen bee, basically passing it down to offspring, right? Or transmitting it to offspring, okay? But one of the most notable characteristics, of course, is that deformed wing. Ultimately, if you can't fly around and pollinate or produce, collect nutrients, essentially, the likelihood of surviving is incredibly low. Okay, good. All right. So these are in terms of viruses. All right, for bees. Going on to another virus that's a very important in Australia, is the, as I have mentioned before, is the Australian bat virus. Okay, and this virus causes a disease called ABL, right? Which is short for Australian bat virus, but it's commonly the disease is referred to as ABL, right? And this virus is very similar to another virus that you probably are familiar with, or you probably have heard of, which is the rabies virus, right? And this ABL, it essentially infects, you know, different types of bat species, right? Fruit bats, microbats, different type of, I don't want to say carnivorous bats, but bats that feed on blood of animals, right? And this disease can be transmitted from bat to human, right? This is an example of the lysovirus here, right? And of course, a bat species, particular bat species. One of the biggest reasons why it's important is because this virus, of course, it can infect humans, and it can provide or give a condition called encephalitis, right? And that's essentially inflammation to the brain, which can be fatal, right? It has a high mortality rate if you get this. Okay, so one thing that it does is essentially inflame the brain, so the brain becomes more swollen and essentially bigger and bigger and bigger, and that itself it provides a whole array of symptoms to the body. Ultimately, I believe if it's too inflamed, it could clog, stop blood supply, and then that's like instant death, if I can say, right? Not instant, but you know, since it does occur gradually. All right, so very important virus. Okay, the bats themselves, right, could show symptoms, so you can, as a researcher, as a scientist, you can kind of detect if they have this virus even before doing blood work and more molecular techniques, right, they can show several symptoms like paralysis, general weakness in the body that they can't even fly. They tend to be shaky, right, or they can have seizures, right? That's one of the characteristics of rabies also, right? But not necessarily all the bats. And this is quite important for this case, right? So they can be symptomatic or asymptomatic, that they don't have any symptoms. Because of that, it's always, you should always be incredibly careful, right? Every bat could potentially be a vector, could potentially be carrying the virus, okay? So they are the reservoirs, they're hosts, right, for this particular virus, okay? A note there, right? Moral of the story in this case, just don't ever touch bats, okay? So mode of transmission, okay? In this case, they replicate in salivary glands, okay? So mode of transmission, because they replicate in salivary glands, bites, scratches, or breaks in this from the bat, and the person having breaks in the skin, open sores, you have a higher chance of getting infected, right? Let's say more commonly of a bite. Let's say you get bit by a bat one way or another, right? That's an immediate transmission from saliva, right? Yeah, so that's essentially how they'll get infected to the new host. The virus gets to the new host, and then from there you get into the bloodstream. But moreover, the virus tends to find its particular receptors in the nervous system, right? Of course, we're talking about encephalitis, where that's part of the nervous system, right? And more particular in sensory nerves and motor nerves, right? So, and then once it kind of establishes itself in the nervous system, its effects is basically inflammation, right? Okay. So something similar to mosquitoes, if I can say, is that it's not contagious. The Ross River virus is not contagious, but infectious, right? You can spread from human to human. And remember, okay, that it's also zoonotic, right? That it goes from animal to human directly, right? Incubation period could vary, right? Depending on the host, also physiology, that it gets infected, from 10 days to over a year. And what are the kind of symptoms that you can show as a human, right? Basically, it's broken down into phases, right? And the first phase, if I can say, you'll have these very general and broad symptoms: fever, headache, nausea, right? You're sensitive to light, winded. You basically get out of breath very fast, very easily, right? And you feel in your overall body a certain kind of internal coldness, they say, right? General body aching, itchiness, right? And some form of numbness, generally in the limbs, right? In the fingers, in the toes. And then once you kind of surpass this phase, that the virus is replicating more and more, and essentially they're establishing themselves in the nervous system, things start to become more critical, right? You start to get general anxiety, unusual eye movements, right? Eye eye is one of the things that's quite particular is that eye is insensitive to touch. Basically, you can touch your eye, you won't really feel that there's a physical touch on getting on your eye, right? Facial muscles will weaken. Remember, this is also influencing muscle nerves, motor nerves, right? Heart respiratory rates will change, right? Can increase. And there's that ultimate third phase, which here you've reached the critical point where you have encephalitis, you have that inflammation of the brain, which earlier instances of encephalitis, you can have paralysis, whole body paralysis, you can enter a coma, and if left, of course, untreated or it's untreatable at that stage, the likelihood of death is pretty high, right? So very critical, very important to understand how this virus works and where you can get it, right? Australian bat Lyssa virus, all right? So here's a little summary kind of, here's a little summary representation just to get a sense, a broad sense of what you have there. Okay, so you have your bat host, then you can get that transmission via a bite, right, to a human, and then you have the whole symptoms in the body. Okay. All right. So this would be a good place to stop if you need to have a break here, right? Because we're going to get into another group of pathogens, which is bacteria. So if you want to stop the video for today, you can do that. All right. So continuing on, now we're going to talk about bacteria. So this is more familiar to what you guys that we've talked about before, right? We're going to talk about several types of bacteria, but some of the most famous ones is tuberculosis, right? And tuberculosis infection caused by a particular bacteria, and so forth, right? So getting into that first one, right, in terms of tuberculosis infection, it's caused by the Mycobacterium tuberculosis. This should be italicized, right? Mycobacterium tuberculosis. You should know these names, all right? And this type of disease, it is common in developing countries, right? And this is a type of bacilli. If you guys remember the shape of bacilli, it's rod shape, right? It looks like a little cylindrical rod, right? And this is particular that it produces tubercles, all right? And I'm going to explain what that is with some pictures here. It's essentially, they're small and round structures that they're made up of macrophages or phagocytes that is produced from the infection itself. They're referred to as tubercles, right? Tubercles. All right. So the some particulars in terms of the infection is that the disease, if I can say, is density dependent. It does tend to spread much faster when you have a high dense population, right? Where we have a high population, and of course, if within the human population you have a lot of individuals that their general immunity is quite low, right? You have a higher degree of spread, right? So again, density, right? So they're in close proximity with each other, each individual person, and of course, being in developing countries where you see this disease more common, right? You do have some kind of recurring situations, right? There is a pattern within developing countries that you do tend to get crammed housing. Hygiene is pretty bad, Cleanliness is pretty bad. So remember, bacteria can lodge onto surfaces, right? If you remember from the experiment that we did, fomites, right? And that can propagate this infection more and more. Hospitals also, the quality of hospitals facilities isn't the greatest in developing countries. That could further promote this this disease, right, to spread, right? So taking a look at this panel here, this figure, tuberculosis ultimately comes in and it's an infection related to the lungs, right? Being that the way of entry, right, and the way of exit is essentially the nose, the mouth, coughing, sneezing, droplets, right? So all these key properties essentially enables that spread here, right? So in terms of transmission, there's two methods, direct and indirect mode of transmission is possible. As you can see here, if a person is coughing, you have these little tubercles, right? These little structures, they're coming in, they're inside these very tiny droplets of water flying in the air. A person inhales these little droplets, they get infected, right? As you can see here, right? You have these little spots here. They get liberated through a sneeze or a cough. Somebody else inhales it. If they're in close proximity, they get their lungs infected, right? Okay, so basically what I just mentioned right now in terms of this pathway. And of course, being in the lungs where you're having an incredibly fast movement, circulation of air coming in and out, in and out, in and out all the time, then the bacteria also move pretty fast, right, to get lodged into the alveoli. Because remember the alveoli, those little sacs where gas exchange is actually occurring, right? So just to go a little bit for lower here. That's it in terms of direct transmission. Of course, you're essentially coughing in front of a person, which you definitely don't do that, right? But, you know, we've talked about indirect transmission already, where if the air droplets are essentially fly off into the air and they pass, you know, a relatively good distance, right? And they collect on dust, on surfaces, they can still get picked up, right? By the wind, whatever means, touching a surface, touching your mouth, inhaling particles that you're just rubbing your skin, right, your fingers on. It's so easily that it can get picked up, right? You inhale that and you get it already, tuberculosis, right? So indirect and direct transmission is possible there. So when going more specifically now, where it's lodging in, basically this figure here, I'm gonna zoom in a little bit more. Let me see here. Yeah, they go inside, you know, the different ramifications of the lungs. You have your trachea, you have in your bronchioles, and then you have the alveoli. You guys remember going in here, ultimately going to these little sacs, right? You have the alveoli. Okay, so once they lodge into that alveoli, you have the bacteria itself, right? Which is that bacilli. And in that bacilli, what's happening is that they'll start to reproduce in there, right? So I'm going to go a little bit higher so you can see more in detail with these following figures here. Right? And of course, bacteria, they undergo binary fission. So that's a mode of reproduction, quick, you know, increase in population, right? Asexual reproduction. And as they start to incubate, right, 2 to 8 weeks later, it's pretty slow in that sense, but white blood cells, right, phagocytes, okay, those cells that could come in and basically undergo phagocytosis, where they can engulf and swallow in, right, these type of pathogens, they come in to that area of infection, right? And what if you remember, one of the particulars about this bacteria is that they can actually evade, right, these phagocytes from eating them up, right? Let me clarify, not just eating, they could engulf them, but they don't get destroyed within the phagocyte, all right? They don't get destroyed within the phagocyte. And the reason for that is because they have a particular type of substance, okay? It's a mycolic acid, which is a kind of substance that has waxy properties, and having that prevents digestion from other substances, right? That the bacteria, that the phagocyte might have, that the macrophage might have. So what happens if you can't destroy it within your body? The macrophages, they work together and they actually encapsulate, right? The clusters of bacteria, little groups of bacteria. And that encapsulation, as you can see here, these little yellow balls are the macrophages that are coming in, and they're essentially creating a little capsule or a shell surrounding the bacteria, trying to contain them. If you can't destroy them, then at least you can contain the situation, right? Creating a barrier shell. That is what you call a tubercle. Okay, so yeah, could have different terms, right? It's also can refer to as a granuloma, more specifically. So these barrier shells, right? This encapsulation, for a period of time, once it gets encased or encapsulated, the bacteria stop growing, right? So you kind of have it, but it's not showing you any symptoms, right? Because in a way, the bacteria get controlled, under control, right? So at that point, it's referred to as latent tuberculosis, right? The bacteria is essentially dormant. There are no symptoms within the host, within a person, right? So at this point, you're not necessarily contagious, all right? You're not necessarily contagious. But what happens afterwards, after a certain period of time, something happens that the shell generally can weaken. That encapsulation of the macrophages gets weakened. They start to wear out. Remember, macrophages are cells, so they could wear out to the point that the bacteria basically can break out of this shell. All right, and at that point, you can become infectious again, contagious. It could be contagious, right? So if the bacteria aren't contained, they basically can burst out, and at that point, the infection is active again, spreading out throughout the whole lungs, giving you symptoms, and you can also be contagious to other people, right? And so forth. So one of the interesting things about tuberculosis in this bacteria is that the World Health Organization recorded 1.5 million deaths by 2018, and they've calculated, if I can say, 1/3 of the human population has latent tuberculosis. So they have the tubercles and they have the encapsulation going on, but they don't have any symptoms, right? So they might not even know, we might not even know that we have tuberculosis, right? And of course, the spread is mainly due to cough, highly contagious, right? Okay. So again, guys, this is another, so those are the defining things about tuberculosis, all right? Know what is the bacteria, the pathogen that creates it, how it spreads, what are the tissues that it infects, okay? All right, tetanus infection. And if you remember, guys, in chapter 12, I talked about all of these in general. Um, uh, what I'm saying now has a little bit more detail, but it's essentially what I've talked about, right? But it's just to ensure that you guys are getting the information down. Okay. Let's see. So yeah, tetanus infection. This is another bacterial infection created or developed by this bacterial pathogen called Clostridium tetani. Okay, and in this case, this is a particular bacteria that is referred to as an obligate anaerobe. Okay, an obligate anaerobic gram-positive bacilli. All right, there's a lot of stuff to unpack there, but mostly what's most important is that it's an obligate anaerobe. All right, so if you know this, if you remember from anaerobic respiration, right, these are bacteria that basically don't use oxygen for their metabolic activities, and actually they grow and multiply in anaerobic conditions or any conditions that doesn't have oxygen. All right, so how does this infection work? You know, what are some characteristics about it? Let's take a look here, right? So first of all, the way that this infection comes in and infects a person in general is usually by the soil, right? Usually by the soil, and the organism, in this case humans, must have some form of opening on the skin, some form of wound, laceration, anything in which the spore, right, or that Latin form of the bacteria can come in, right? Usually walking barefoot on soil, you know, and whatnot. They typically like to thrive in deep wounds, in deep tissues, right? Deep muscle tissues. There's not much oxygen in general flowing, circulating within our body in these particular tissues, right? So they're generally hypoxic, right? They have very low oxygen levels compared to like the blood or the lungs, right? So one thing about this infection is that the spores, right, they will germinate in hypoxic conditions like deep wounds. Okay. So, and as I go along, you could basically see these little visuals here where you have these bacterial spores, not just soil, right? They could also be found on rust. It grows fairly well on rust, right? Other animals, right? And one way or another, if you get a puncture here, right, you get some form of entry, right, by a skin wound, you're gonna get it if the soil is contaminated, right? Some other ways, like stepping on a nail or getting bitten by a dog, and so you can get this infection. Okay. All right, so after you basically get this spore form and it starts germinating, the bacteria will tend to replicate, reproduce. I want to say more. Replicating is more for viruses. Right, it'll start to multiply, reproduce by binary fission. You guys know already, a mode of asexual reproduction, increasing the population numbers. So as this is happening, right, it's spreading around, especially if it gets, you know, spreading around different tissues. It goes through the bloodstream, circulating through different areas of the body. It could get into the nerve cells, or that's essentially its go-to tissue, the nerve cells, right? And one thing that's particular about this bacteria is that it produces a toxin, okay, that's called tetanospasmin. Okay, tetanospasmin, and you should definitely remember that, right? Tetanospasmin is basically a toxin that affects the nervous system and the neurotransmitters, right? So what is particularly interesting or important to know, right, as a researcher, as a medical doctor, is that this toxin cannot be neutralized by an antitoxin, right, or some form of anti-serum, if I can say, right, that we know of. What happens for this particular toxin is that the bacteria is growing, is producing this toxin, it's secreting it. This particular toxin blocks, okay, it blocks the release, okay, of inhibitory neurotransmitters. All right, so what does that mean? Inhibitory neurotransmitters are those that essentially stop a nerve impulse or inactivate a nerve impulse, right? I don't know if you've seen with Mr. Naughton, but there's inhibitory neurotransmitters and there's excitatory neurotransmitters, basically stopping a nerve impulse or um, uh, promoting a nerve impulse, right? So in this case, you have a toxin that blocks the inhibitory neurotransmitters. So if you block these inhibitors, you're always going to have, and you have the excitatory neurotransmitters active, you're going to have always a signal being produced active. All right. So because of that, these particular signals that are occurring, they are related to the muscle. And you essentially have uncontrolled muscle tension, right? And uncontrolled muscle spasms, moreover, of particular muscles, which are called the skeletal muscles, right? And if you guys remember from the pictures that I showed you before in the previous chapter, it's one of the defining features is lockjaw, that basically the jaws, you can't open your mouth, your jaw is locked in position because of that excitatory response. Some of your arms, they tend to be your fingers tend to be curled up, even your arms are kind of close to your body, your back is bent backwards, right? Your back is bent backwards, right? And you generally have symptoms related to the muscle, okay? And you get these postures, if you remember from the picture, I should have put it here, that is quite agonizing, which is referred to epistotonos. I don't think you need to know that, though. All right. But moreover than that, than just the muscular system, you can get very big, critical situations where you have problems with the respiratory system, right? The diaphragm, which is that little tissue that's below the lungs that helps it expand and contract and rest, right? Doesn't function properly, right? Muscles are obstructing the airways. So you have essentially difficulty breathing, severe difficulty breathing, right? And in situations where the neurotransmitters is so intense, just from the power of your muscles, you can actually fracture some of your bones, right? That's how severe the situation could be with this type of disease. Okay. Again, so symptoms, mode of entry, always be familiar with these things for each pathogen. This disease, again, is infectious, but not contagious. All right. It's important, it is likely as a question, you could have name some diseases that are infectious, but are not contagious, as opposed that are highly contagious. So make sure you kind of make a note on that. All right. So that's in terms of bacteria. Wait a second. yeah, I'm probably gonna cut this part off here. I'm gonna leave the video for now, and I'm gonna make a part 2 of the video. All right, guys. Stop sharing and